This is a recording for the Biotechnology Lecture for Monday, February 27th. We are starting on slide 32 of Chapter 8, and we will extend on to Chapter 9. However, Chapter 9 will not be on Exam 2, which is next Friday. Again, when we're looking at familial relationships and DNA profiles, one thing that is done is to test mitochondrial DNA. Many times nuclear DNA is not available after cells degrade, or perhaps you don't have a cell sample and instead you have a sample of a hair shaft. The mitochondrial DNA will remain, so that's really your only choice in terms of identification. These are can't be analyzed by RFLP or STR because the mitochondrial genome is so much smaller. It's only 16,000 base pairs, and so an RFLP analysis is probably not going to be significant enough to identify an individual. This is for older samples that once the nuclear material has degraded, this would be hair, bones, and teeth, where mitochondrial DNA are left behind. And the mitochondria are inherited from the mother only, so the inheritance pattern is very easy to analyze because you have the same record and a very pristine record of your mother's DNA. And there's only a very, very slight amount of change in the mitochondrial genome, about 1% every million years, due to some type of random mutation. Okay? And here we see up on top, Part A, mitochondrial DNA for the target mother here. They've done a restriction fragment length analysis, and it gives a particular pattern. And that's the same pattern that you would see in all the children of this particular mother, regardless whether male or female, or regardless of who the father is. Here's the mitochondrial DNA for another female. Females are circles, and you see an unrelated pattern. Okay. Some of these will be similar because mitochondrial DNA for humans is well conserved. But you will see some slight differences. Now, if we see male and female mitochondrial DNA, the female is denoted in blue, and she will donate the mitochondrial DNA. Mitochondrial DNA are in the egg and not in sperm. And so she will donate her mitochondrial DNA to all the children, regardless of male, female. Here's another male, female, and male. Okay. Now, the resulting females here, only the females, the daughters, will pass down the same mitochondrial DNA to their children. Okay, so their children are all labeled in blue. This is his grandchildren generation, and so all the progeny have the same DNA pattern. All the progeny here will have the same DNA pattern. However, for the males, for the sons, who mate with other unrelated women, then the patterns will not be the same because the patterns are going to be inherited from the female and not the male. For males only, you could do a Y chromosome analysis. The Y chromosome goes directly from the father to the son. Like the mitochondria, it is well conserved. However, it has nothing to do with daughters because women do not have a Y chromosome. So you can trace relationships among males and analyze biological evidence. If you have male suspects, then you can do a Y chromosome analysis. In terms of non-human DNA analysis, in order to verify the authenticity of different lines of plants, especially things that are valuable like ginseng. Ginseng has a $3 million market in the U.S. alone, which is actually pretty small. The markets are much larger in Southeast Asia, where ginseng is more popular. And there are two major herbal products that are referred to as ginseng. One is native to North America, the other is native to Asia. The Asian ginseng boosts energy, the American ginseng calms nerves. And in order to tell the difference between these strains, then DNA analysis has to be done on the individual plants to identify, did this come from North America or did it come from Asia? Same thing in terms of winemaking. Cabernet Sauvignon is a type of wine, and it comes from grapes that are grown in a particular region in France. And hybrid grapes, which are not purely Cabernet Sauvignon grapes, are considered inferior, and they're excluded from having the prestigious distinction which d'origine contrôlée in France. And so DNA evidence is used routinely to verify that the grapes are Cabernet Sauvignon. So a small sample of DNA is obtained from the grape, and then they can determine whether it's Cabernet Sauvignon. Other varieties would be Cabernet Franc and Sauvignon Blanc. In terms of collecting samples from animals, they could use this in order to identify poachers who have illegally hunted bear, and they have been able to do this and matched actually the crime scene where the bear was killed in a den. It's illegal to kill a bear in a den in Pennsylvania. You can kill them out in the open for self-defense. You can kill them out in the open for hunting, but you cannot go into their dens and kill them. And so they were able to collect blood samples from the crime scene, match the samples with the bear that was killed, and convict the hunter of the crime. Some artifacts and memorabilia are tagged with DNA. Footballs don't have their own DNA. They're not, they're made out of plastic or pigskin, but they don't really have a particular DNA. So a DNA tag will be placed in the item itself, and that will be used to authenticate that this did directly come from, Super Bowl 2003 or paraphernalia and trinkets from the 2000 Sydney Summer Olympics. And that concludes Chapter 8. We will move on to Chapter 9, which is on bioremediation. Okay, so first of all, we're going to figure out what bioremediation is. Obviously, it's biological methods that you can use to remediate or clean up contaminated waste sites. We'll cover the basics. We'll look at different sites that need to be cleaned up and give an overview of strategies used for cleanup of sites. We'll talk about how genetic engineering is used. We'll do case studies in bioremediation. And then we'll talk about future challenges, how bioremediation is used today and how it will need to change in the future in order to clean up the legacy waste sites, specifically in the United States. So bioremediation, you can split this into two terms. One is a little more general for the process of breaking down contaminants, and this is called biodegradation. It's the use of living organisms to degrade chemical compounds. Now, when we talk about it in terms of cleaning up environmental sites contaminated with chemical pollutants, then we talk about bioremediation. So biodegradation is for any degradation process. Bioremediation is limited to contaminated sites. It is important because we have a lot of environmental messes, and environmental chemicals influence our genetics. Not only do they influence our genetics, but they influence our bodies in general. And so we need to be very, very careful in terms of what chemicals have been disposed and what exposures are doing to human health as well as environmental health. There is concern about short-term exposure, obviously, but also long-term exposure can cause certain forms of cancer as well as birth defects. And we produce a lot of hazardous waste in the United States. Over 200 million tons of hazardous material is produced in the U.S. each year. So in 1980, under the Carter administration, Congress implemented what's called the Superfund program. The Superfund program was a large base of tax dollars initiated by the U.S. EPA in order to counteract the careless waste of dumping of waste in the most contaminated sites across the United States. And the purpose was to locate and clean up these hazardous waste sites using government funds, as well as figuring out which chemical companies, which entities were liable for this contamination and holding them accountable as well. And it turns out that 1 of 5 Americans lives near a polluted site treated by the EPA. We live close to Iron Mountain Mine, which is a mine about 9 miles out of Reading, and it is a Superfund site. It has been under environmental cleanup since the 1980s. And in the 25 years since Superfund began, this is an old figure obviously because it's been longer than 25 years since 1980. The EPA has cleaned up about 700 sites. However, as many as 200,000 sites still need remediation, so we still have a long ways to go. And the cleanup cost is in the trillions of dollars. Obviously, this eclipses the federal government budget, so there's no way that these sites can be cleaned up in a timely fashion. Also, there's the Environmental Genome Project, and this is to understand the impact of environmental chemicals on the genome and to find genes sensitive to environmental agents, to learn more about those genes responsible for detoxification, and then identify SNPs. These are single nucleotide polymorphisms in populations that may be more susceptible to human health concerns due to environmental exposure. This will also generate genomic data, and this will be able to tie specific genomes and SNPs to different environmental contaminants and understand how environmental conditions can influence disease. So why use bioremediation? Mostly our emphasis is to take environmental contaminants and convert them into something harmless. So rather than moving the waste and locating the waste in a separate facility where it still is toxic, we convert the waste into things like carbon dioxide, chloride ion, and water, as well as simple organic molecules that can be absorbed by the environment. Bioremediation processes are generally cleaner, less harsh than traditional waste treatment processes, and they can be conducted at the site of the pollution, which saves transportation costs. It's very difficult to transport toxic waste, and so if we can clean up the site at the source without moving the waste, then we'll save a lot of money. Okay, so what needs to be cleaned up? Almost everything. Oil, water, air, soil, sediments are the most common. And each presents its own type of complexity. Each molecule that needs to be biodegraded has to have a different biochemical pathway. And also the site conditions, whether it's soil, air, or water, the degree of saturation of the water in the soil, the composition of the soil, all present complexities that are unique that have to be dealt with on an individual basis. And pollutants enter the environment in many different ways. You can have a tanker spill or a truck accident. A lot of chemical tanks are ruptured and leak. Now, most industry experts recommend double-walled tanks instead of single-walled tanks to prevent spillage, so you've got at least an extra layer of containment, and then release of pollutants in the air. This also depends on the location of the accident, how much chemical was released, and the duration of the spill. Okay, and here are just some examples of exposures. Here we've got a leaking chemical tank that is pouring chemical contaminants directly on the soil. You can have underwater tanks that do the same thing and leak contaminants into the soil subsurface. And then the soil subsurface is divided between the unsaturated zone, which is above this line, and groundwater, which is below this line. This groundwater is an aquifer. Aquifer is just an underwater reservoir, okay? And as these chemicals leak into the aquifer, then they're contaminating the water source that nearby homes are using as their well water, okay? And so not only are we looking at the impact of the environment, which any of these spills would impact the environment, but we're also impacting human health because these chemicals are contaminating an aquifer that's responsible for residential water. And the biggies, the chemicals that we're most concerned with are carcinogens and mutagens. Carcinogens are compounds that mutate DNA that cause cancer, and mutagens or teratogens are compounds that affect the gametes and will cause birth defects in the next generation. You need to be very, very careful with exposures to pregnant women, where the somatic DNA of the developing unborn child can be mutated. Also, any mutation of the gametes is going to pass along birth defects. Okay. Then other chemicals cause skin rashes, like I said, birth defects, and then including those that would poison plant or animal life. As plants are poisoned, then that leads to economic disaster, leads to the inability of to detoxify the environment because the plants are so important in detoxification. And then animal life then basically accumulates in the food chain. You can cause animals to go extinct because of environmental pollution. So all of these concerns come into play. And here's a long list of contaminants. Each one of these has to be handled individually. Some of these, like benzene, are organic contaminants, and they're more difficult to break down in the environment. Others are heavy metal contaminants, like chromium. What are some others? Trace metals, arsenic, cadmium, chromium, copper, lead, mercury, and silver. And remember, a metal, when it's remediated, you can't change a metal into a non-metal. It's atomic. So the metals tend to produce a more of a thorny problem because they have to be remediated and taken out of the environment. You can't just convert them into innocuous chemicals, but they have to be accumulated and either rendered insoluble, so they don't leach into water supply, or they need to be collected and taken out of the contaminated site. So basically, if we break down these particular chemicals, we're looking at metals and organics. Those are the worst of the worst, and they are the ones that need to be cleaned up first. So fundamentals, microbes convert chemicals into harmless substances via aerobic or anaerobic metabolism. And this involves oxidation and reduction reactions. Of course, oxidation and reduction are coupled. Oxidation when you remove one or more electrons from an atom, and reduction when you gain electrons, an atom gains electrons. Okay, so when you start out with an electron donor and an electron acceptor, then the electron donor becomes oxidized, it lost its electron, and the electron acceptor becomes reduced because it gained an electron. Okay, and in terms of aerobic biodegradation, the terminal electron acceptor is always oxygen. So here's benzene. It will donate its electrons to oxygen, and it will be converted to carbon dioxide and water. And then biomass, which is just an increase in the number of bacteria. In anaerobic biodegradation, here's toluene, and it's being degraded in the presence of nitrate as the terminal electron acceptor. And nitrate is not the only terminal electron acceptor in anaerobic biodegradation. There are several different electron acceptors. Nitrate is just one of them for denitrifying bacteria. And in this case, you get carbon dioxide as well as nitrogen gas, water, and biomass. Okay, and you're looking at microbes that can metabolize a contaminant. So you're trying to identify a specialized source of microorganisms. And these are used as the tools that are going to convert the very complex biodegradation of these compounds. Many may use both aerobic and anaerobic processes. So you have aerobic bacteria, which are acting separately from anaerobic bacteria. Okay, and to show the different zones, to start out with, there is oxygen in soil, and here is groundwater. Groundwater is just soil that is saturated with water. Okay, and out of the contaminant degradation zone, the oil is basically aerobic. Okay, or I'm sorry, the groundwater is basically aerobic. And inside the contaminant degradation zone, here is the contaminant source. Now, the first thing that happens in these degradation reactions is aerobic metabolism. So we have benzene that is being converted in the presence of oxygen as a terminal electron acceptor. It's converted to water and carbon dioxide. And then the next region to be stimulated are nitrate-reducing anaerobic organisms. Okay, in this case, nitrate is reduced to nitrogen gas. Carbon dioxide and water are also formed. And then the next region, as we are getting closer and closer to the contaminant source, are iron-reducing organisms. They reduce iron 3 to iron 2. They also produce carbon dioxide and water. Next are the sulfate-reducing organisms. They convert sulfate to elemental sulfur. And carbon dioxide is produced in water in the degradation of benzene. And then finally, because carbon dioxide is ubiquitous in soil, carbon dioxide is present because of the degradation of limestone, which is carbonate. Okay, and this is converted to methane gas and carbon dioxide in the presence of carbon dioxide. Okay, and here's the contaminant source. So I want you to pay very close attention to this. The first reactions that occur in the groundwater when a contaminant is encountered are aerobic. Then after the oxygen is depleted, then you get a zone of denitrification where nitrate is the electron acceptor. After nitrate is depleted, then you get an iron-reducing zone where iron 3 is the electron acceptor. Once iron 3 is depleted, then you get sulfate-reducing organisms. Once sulfate is depleted, then you get methanogenic, these produce methane organisms in the presence of carbon dioxide. Okay, so know those terminal electron acceptors, know what we call the bacteria, and also know the order in which contaminants are degraded when they're encountered in the groundwater. Mostly we try to use indigenous microbes. Microbes in a contaminated site have adapted to the presence of the contaminant, and many are there because they have the ability to degrade the contaminant. They're often isolated, grown and studied in the lab, and then released back in large numbers, or they are completely in situ. Some examples are Pseudomonas and E. coli are ubiquitous in soil. You find them everywhere. And so, especially Pseudomonas, so they can be bulked up, reintroduced into the contaminated environment and degrade the contaminant. And the quest for new activities, things that can degrade contaminants that are more difficult to degrade, is an active area of research. Scientists are always looking at rich sources of microbes like contaminated soil or sewage sludge to see if decontaminating organisms can be isolated. One specialized bacteria was found in California's own Mono Lake. And this particular bacteria metabolizes arsenic and incorporates it into DNA. Arsenic is an element, and so it can only be bioaccumulated. It can't be converted to something that's non-toxic. But the organism itself, once it grows and incorporates the arsenic, may be collected in order to reduce the arsenic in the environment. There are also algae and fungi that are capable of biodegradation. The fungi Phanerochaete chrysosporium and Phanerochaete sordida are used in degrading contaminants associated with wood treatment. This would be like creosote, pentachlorophenol, and other pollutants. Wherever you see a wood treatment plant, it has a particular odor associated with it. Usually that's creosote, and creosote is very difficult to degrade, but it will degrade in the presence of these fungi. Asbestos and heavy metals are accumulated using Fusarium oxysporum and Montiarella hyaline. And also fungi are used in composting, sewage treatment, and at solid waste and wastewater plants. One group of contaminants that's difficult to degrade is called polychlorinated biphenyls. These were historically used in electrical transformers, so you find them a lot in the environment associated with electrical power stations. And these were previously thought to be recalcitrant. It was thought that they could not be biodegraded. They can, however, you have to use the proper source of fungus. There is a bioremediation genomes project, or a series of programs. And this is where scientists are looking at the specific genes and organisms used for bioremediation. And once novel pathways can be identified, then these pathways can be genetically conferred to indigenous bacteria for the cleanup of soil and water. So we're using genetic engineering to help strains that are traditionally used for biodegradation. Okay. And here are some genomes for different organisms that have been identified for treatment. One contaminant that is very, very difficult to degrade is perchloroethylene. And so you see it here twice, perchloroethylene for Dihalobacter restrictus and Dihalococoides ethogenes. These have been isolated. Dihalococoides ethogenes was isolated from a waste treatment plant, and it will reductively dechlorinate perchloroethylene to trichloroethylene to dichloroethylene to vinyl chloride, and then finally to ethylene. And just some basics. It turns out that when you have petroleum products, like an oil spill, like the Deepwater Horizon oil spill that happened in the Gulf of Mexico, all you need to do is provide oxygen. Okay, so rather than providing pure oxygen, air is cheaper. So these zones are aerated. This causes native bacteria to grow. You also add fertilizers for these other nutrients, nitrogen, phosphorus, carbon, and hydrogen. And this is sort of a recipe for petroleum hydrocarbon degradation, and it is very effective. You can also use phytoremediation. This is when terrestrial plants are used to clean up the soil, water, and air. And plants will naturally take up toxic species. You find these plant species like ferns or parrot feather in wetlands, and they will hyperaccumulate metals. They will detoxify organics. Some of these also include poplar and juniper trees. Let's go back. Alfalfa and grasses. Sunflower plants were used to hyperaccumulate cesium at the Chernobyl nuclear plant disaster. Water hyacinths that are found in wetlands were used to remove arsenic from the water supplies in Bangladesh and India. Okay, and so the roots absorb the pollutants. Okay, and typically these are metals, so they can't be completely detoxified in the environment, so they have to hyperaccumulate in the plant. Then the plant has to be removed for disposal, and the disposal, you have to be very, very careful that you don't recontaminate the environment, especially with airborne metals that would, if you burn these plants, would escape from the stack. So chemical pollutants are taken through the roots of the plants as they absorb the water. The plants may use enzymes to degrade the chemicals, or in the case of metals, just hyperaccumulate. So in the case of metals, the chemicals can be concentrated in the plant cells. The plant serves as a sponge, and the contaminated plants are removed from the site and treated as waste. Okay. High concentration of chemicals will kill most plants, so these will work better at areas of low concentration. It also tends to be a slower process, so you cannot reclaim contaminated land very quickly. A lot of times these plots are maintained for a number of years, and the plants are cycled. You grow plants, accumulate the contaminants, harvest them, then plant new plants, and repeat the process several times. Plants naturally move carbon dioxide through photosynthesis. And so it's very interesting that we have greenhouse gas problems, which is an accumulation of carbon dioxide. So one way to remove carbon dioxide from the environment is to plant more plants. And plants like the black cottonwood, which is a type of poplar, which will take up very, very high levels of carbon dioxide. It has a high rate of photosynthesis, are very promising. These are genetically modified, so they'll even increase the photosynthesis rate further. It's also very eye appealing, but you can only treat surface layers, so around 50 centimeters deep can be treated. Like I said before, the cleanup takes several years. Whoops. Okay, so we're transitioning to cleanup sites and strategies. Okay, and here are some things that need to be considered when you approach the cleanup process. Are the chemicals flammable or an explosion hazard? I encountered a site which was a munitions site where our munitions were fired at a army base, and there we called it boom soil because there was enough TNT that was dissolved in the soil that made the soil explosive. Okay. Do the chemicals pose a threat to human health? And so do you need to take special precautions for cleanup workers? How was the chemical released? Was it a single incident, or was it like a leaky tank that leaked over a number of years? I've worked with leaky tanks that have leaked over periods of 40 to 50 years, and so the contaminant is much more widespread, and it has gone further down into the soil and into the groundwater. Whereas a single release that is a small release, you may treat differently. Where did the contamination occur? Soil, air, water. Is it at the surface of the soil? Is it below the ground? Is it in groundwater? So does it affect human water sources? How large is the contaminated area? Okay, and so for soil cleanup, you can do it ex situ. Ex situ is just outside of the site. And so you have to take the contaminated soil and you have to move it to another area for treatment. In situ leaves a contaminant in place. And this is the preferred method because you don't have transportation costs and larger contaminated areas can be treated in one time. Usually you're looking at pre-existing microorganisms in the soil and water. So you're stimulating microorganisms, you're not adding new ones to the environment. The microorganisms that are already in the soil and the water have adapted to the presence of the contaminant, and usually several microbes within the microbial community will directly degrade the contaminant and reduce them all the way to carbon dioxide in water. So with in situ bioremediation, if you're trying to stimulate aerobic degradation, then you may use a method called bioventing, where you pump air or you pump hydrogen peroxide, which when released will produce water and oxygen into the contaminated soil. And you may add additional nutrients like fertilizers that will add nitrogen, phosphorus, carbon, and oxygen to the environment to stimulate growth and degradation activity. But aerobic may not be the best solution. This aerobic bioventing, you need more porous soil like sandy soil, and this allows the microorganisms and the fertilizing materials to spread rapidly. But if you have soils that are high in clay content or have dense rocks, then it's more difficult to distribute the oxygen and the nutrients. So this may not be the best solution. Okay, and bioventing usually is for very quickly degraded contaminants like gasoline. Can be done very easily with bioventing, but those chemicals that take a long time to degrade, like chlorinated hydrocarbons, you need to pick a different strategy. Ex situ bioremediation, although it's expensive, can be faster and more effective. You take the soil and add water, so you create a slurry. You mix the soil with water, fertilizer, and you put it in large bioreactors. And this is good for smaller areas of more concentrated contamination. Then you can do this ex situ in a solid phase. And there are three different types of solid phase remediation that you need to know: composting, land farming, and biopiles. Okay. And so with ex situ bioremediation, you're taking the contaminant, you're excavating it. You could go to a slurry phase bioreactor where this is in a liquid, and you mix the liquid with nutrients to get rid of the pollutants. Or you could go with composting, where you stimulate different activity. You bulk up the contaminated soil with hay and other agents that create pore spaces that stimulate aerobic activity. In land farming, you spread out the soil into a very, very thin layer, and then you leach out the contaminant out of the bottom. Okay, so you have some type of filter underneath the soil, and you collect the leachate, which is going to have the contaminant, and then that can be either concentrated and stored or further treated. And then with biopiles, you do the opposite. You create a soil pile, and you let the contaminant volatilize. So many organic contaminants will vaporize in the presence of oxygen, and you just collect the vapors, and the vapors are then treated. And here's an example of biopiles. Okay. And here's a slurry phase additive. This is just very simple bioremediation that everybody does that has a septic tank. If you live on a septic system rather than having like municipal water treatment, then you can add this and it will break down the contaminants and break down the sludge that is in your septic system and prevent it from backing up. Okay. And at this point, I am going to conclude the lecture on bioremediation. We will start up with this lecture after the exam, so this will be the subject of the lecture starting a week from Monday.